think let's go ahead and get started. Welcome everybody to Hutch at Home, our series that brings science into your home from Fred Hutch and from some of the partner organizations that we work with. I'm Jeannie Chowning, the Senior Director of Science Education at Fred Hutch, and I'm really excited today to introduce you to Ricky Padilla Del Valle, and he's going to talk about It Takes Two to Tango, the evolution of L1 transposable elements in the human genome.
Thank you so much.
So just right before we start, this is a talk geared towards science teachers and their students. And so if you are a practicing scientist and want to ask more detailed questions, please wait until the end. And Ricky said he could stay on a little bit if you want to ask super technical questions. But just remember that our primary audience is science teachers and their students. If you could please mute yourself if you're not talking, especially if you're coming in from a phone. Feel free to add your questions into the chat though, and then when we get to some good stopping points, we can look at those and address those. And please complete our feedback form at the end of the talk, and I'll be sure to put the link into the chat for you. Okay, so really excited, as I mentioned, for this talk. So Ricky is a molecular and cellular biology graduate student through the Fred Hutch University of Washington MCB program. And he's currently at the McLaughlin Lab at Pacific Northwest Research Institute. And I believe that's where you are today in your background, right? So I was really, from the little that I heard of this talk when we were planning it, I think it's a super interesting topic, and I'm really looking forward to hearing this talk. So I'm going to turn the screen over to you. And so go ahead and
okay, just a second. Yes, I am sharing my screen. Great. So I'm Ricky. You can refer to me as Ricky. My pronouns are he, him, they, or them. I Jeannie said, I'm a second year grad student in the molecular and cellular biology program at UW, and I work in the McLaughlin Lab at Pacific Northwest Research Institute up in First Hill. So for today's agenda, I'm going to talk about me and my academic journey. And then we're going to go into more science, like what is the biology of transposable elements, a special one, which we call line one, and human evolution. And then we're going to mention some of the research questions and the approach that we take to answer those, and summary, acknowledgements, and you can ask me the questions that you have. So I was born in Puerto Rico. I was born and raised there. I moved from there to here, to Seattle. So Puerto Rico is a US territory, and it's between the Dominican Republic and the British Virgin Islands. So my first language is Spanish, and I am part of a family of 5. Both of my parents are teachers of elementary school and middle school, and they have been for like 20-ish years. So I get it in some ways. I think teaching is a superhero work, and one of the reasons my parents are excellent educator as of today is because of programs like Science Partnership Education Program, the Science Education Partnership Program. So I want to thank you, Janine, and every one of you for having me today. So in terms of my academic journey, as a kid, I was always really into my surroundings. Like I was either climbing around, finding rocks, trying to like camp and see the stars and all these things. I was never like computer kid, but more like just having the rocks in my hand was like amazing. So when I was in middle school, I decided I want to invest in it in a more like specialized education. So I went to the high school, which is specialized in STEM, is a boarding school in the island, and then like it's a public school, so you don't have to like pay any money, but you have to have a test and all these things. And the good thing about this school is that in addition to finishing in 3 years, you are like they gave us like all these specialized courses like particle physics and biology. And those 3 years for me really formed the core of my interest in science research. So after that, I went to college at the University of Puerto Rico, and I majored in microbiology. My main research work, was in environmental microbiology. I wanted to understand microbial communities, and I wanted to understand how the knowledge that we gather from them could be utilized for their potential in like resolving problems in the clinical environment, like antibiotic resistance or the contamination of soils. So in the summers, I did research internships because I was super interested in not only like understanding microbial communities as a whole, but like how can a bacteria, how can an organism can actually degrade contaminants, and how can like it can resolve that, and the field that works more into that is like molecular biology and like what are genes and proteins and how they interact. So I went to UConn and studied some like gene regulation, so cycle arrest. And then I went to McDonald Genome Institute for the summer and studied some breast cancer and nuclear communication. And then the last one before I graduated college, I went to the Hutch, and I worked with Dr. Michael Emmerman, and we study flu restriction and evolution. And that was it for me. Like I knew I really like Seattle, but the research specifically, it's amazing how these viruses, these tiny entities with so like low resources, can parasite and endanger like entire populations for millions of years. So I decided that I wanted to come here and study more about that. So I joined the University of Washington, the molecular and cellular biology program, rotated in the labs that were focused on proteins and evolution and viruses. And then I joined the McLaughlin Lab. And you're gonna hear more about what we do here. So further on in my future, I think I wanna complete a postdoc and then to like specialize my studies, but my goal as a career, I think I want to be a science educator or like a full-time researcher, depending on which one I enjoy more at the time. So that's there. Okay, so that's my who I am in my academic journey. And what brings me to you today, it's my research here at the McLaughlin Lab. So I bet that some of you, like me, have some hair growth in the last months. Yeah, this is like a lot. So in addition to hair growth, the bone content, lactose intolerance, your color of the eyes, muscle mass, and your hair stamina, all of those are defined by proteins that are produced as a result of reading coding DNA. But when we look further into the human genome, the portion that makes up all that you can see here is actually pretty small. It's like about 1.5% of the genome. So it's like, have you ever like read 15, 20 research articles and then you end up with like one sentence? It's pretty much like that. There's like just a tiny part of the genome which is responsible for all that I mentioned before. However, two-thirds of the genomes are just transposable elements. And these are also genes, but these are really special. I'm going to tell you more about that. So transposable elements are, or TEs, they are DNA sequences that have the ability to change their position within your genome. What that means is they can literally jump into a new place, into a new gene, into a new chromosome, and that's um, the amount of transposable elements that we have in our genome is pretty concerning, because that means that if you take a sample of your cells right now, of the DNA, the genetic information, you have all these jumping pieces of DNA hanging around, but somehow you're okay. Somehow we're like walking around and fine. When these transposable elements were first described, it was in maize. Barbara McClintock described them as jumping genes, and they are directly responsible of the differences in color pattern that you can see in kernels. And the other amazing thing that Barbara McClintock brought up when she discovered this and she described this is that our genomes are not stationary entities, but they are flexible. They can alter. And her work was like amazing. She got like Nobel Prize for that. And after that, understanding transposable elements has been an ongoing research work, because it's really hard to track and understand something that is always changing places and like moving around. So okay, so there are like two classes of TEs. They're DNA transposons. They are known for cutting and pasting. So what they do is that you have a DNA transposon that is in an old position in your genome, and then it cuts and pastes into a new place. And then it cuts and pastes into a new place, into another place. And sometimes these transposons get mutations that stops their ability to cut and paste again. The way this looks in the genome is you have your chromosomes, and then this DNA transposons jumps into a new place and then jumps into a new place, leaving that previous space behind empty. And the other class of transposable elements are retrotransposons, and these are known because they copy and paste. So what they do is that from DNA in the old position, they make a copy in the RNA form, and then via reverse transcription, they insert themselves into new genomic space, which is why they're called retrotransposons, because they go from RNA intermediate, from RNA to DNA again. And these as well can accumulate mutations that will make them will stop their ability to jump again into a new place. The way this looks in the genome, again, you have the retrotransposons, and then you will go to new places, but the difference is that it will stay behind. It will stay in the old location too. So in the McLaughlin Lab, we studied this particularly interesting and special retrotransposon called line 1, or the long interspersed element 1. And the reason why it's so special is because line 1 is the only autonomous and measurably active TE in humans. By autonomous, I mean that these transposable elements is not only does not only jumps around, but it guides itself. It has genes that codes for proteins that move the whole entity around. Because of that, because they're autonomous, they have also been described as selfish, because their sequence only encodes for its own mobilization. It's like they only care about themselves, in essence. So if we look deeper into what line 1 looks like at a molecular level, it's an approximately 6 kb, 6,000 bases long, and it has 2 genes, ORF1 and ORF2. These 2 genes, after transcription and translation, they encode for these 2 proteins, ORF1P and ORF2P. And these 2 proteins, what they do is that they recognize line 1 in the RNA form, and they create this complex with a really long name called ribonucleoprotein complex. And this is the unit that goes back into the nucleus and reinserts itself into a new genomic location. So up until now, I told you that we have this huge amount of transposable elements in the genome, and that there are different ways that they can jump. They can cut and paste, or they can copy and paste, and that the long interspersed element 1 or line 1 is really special because it copies and pastes in an autonomous way, using its own protein, its own machinery. These TEs, when they were discovered, it was mainly the motivation behind the observations that led up to this discovery was color changes in kernel, in corn. But in humans, this manifests differently. If you think about a piece of DNA jumping in your genome and possibly getting to the 1.5% of coding protein that is responsible for making hair or for your heart stamina, there's a huge possibility that it jumps into a gene that can generate disease. And this happens. There are a lot of cancers and a lot of genetic syndromes that have been associated with line 1 insertions. So the host, or humans, we have a mechanism of defense. So no worry, the reason why we are alive and we're fine and we're walking around is because our genome also codes for proteins that can target line 1, jumping around and stop that from happening. So these proteins are called restriction factors because they restrict and they can obstruct line 1 replication, and they can target any step of this cycle. They can target when line 1 goes back into the nucleus, or when the proteins are identifying line 1 in the mRNA form, or when these proteins are formed, or the expression of line 1 itself at a genetic level. But we also know that if we have mutations in host factors, that results in disease. So there are 2 outcomes that are harmful: that we have expression, that we have activity of these jumping genes, and that if we lost control, if we lost regulation of that activity, that also results in disease. In our lab, we're interested in understanding how this interaction happens, how line 1s and humans interact with each other, and how that happens over time. Line 1 is not only autonomous and selfish and like the only measurably active in humans, but is the most successful transposable element family in evolution, meaning that it has been present in mammals for more than 170 million years. So this transposable element is really good at changing and adapting to whatever environment or context or host is in it. And we humans well, like known for being a pretty resilient species, but the worry, the danger that this pathogen, this selfish element, can become detrimental to us at some point. And there are many examples where that is true for a lot of cancers and genetic syndromes. So one hypothesis that explains how these two entities evolve is explained by the red queen hypothesis, and this refers to a passage from Alice in Wonderland. It says, it takes all the running you can do to stay in the same place. And what this red queen hypothesis refers to in our case is that it proposes that a species must constantly adapt, evolve, and proliferate in order to survive while in conflict with another species. And in this case, it's not a species against another species, like a host against line one. Human restriction factors, human mechanisms of defense against the jumping of this transposable element. So what happens is that the hosts have evolved, we have evolved to regulate line one. But if there's a mutation event in line one that allows for it to evade restriction, to evade regulation, then this line one will become active and will potentially generate disease. So these pressures the host to evolve and change in order to regain control and claim that winning state. So this has always been referred as like a conflict, an arms race, but I like to call it two to tango, because if you have any experience going to ballroom, these squares are really important. You have to like do square like paces to like, and they're like these two entities that are always like at each other, trying to understand, trying to like to be with each other, which is why I call it two to tango. So in general, these hypotheses and what we know about the interaction of humans and line ones are what motivates the research that we do. Specifically, I want to understand how line one could evade the host, and this could and therefore generate disease in a new way that we don't understand. So what you see in the top is like host factors that I showed before, and that's interaction with line ones in the genome. And the main question behind our research is, and my research specifically, is how line one has evolved in response to this host control. We have different experiments, different methods that we can use to address this question. And I'm gonna mention a couple and then discuss some results that I have so far. Okay, so we can use computational biology and phylogenetics. In computational biology, with computational biology, we can access the sequences of all these line ones and all of these restriction factors that are in humans. And we can ask questions like, where in the genome are these line ones located? Does that matter? How do they look? How many are there? With phylogenetics, we can not only use that information of computational biology, but like ask, how is line one as a gene evolving in the human genome? Is it constantly adapting like the hypothesis says? Is it dying? Is it like not, is it being like beaten by the host? We can also use molecular biology and cellular biology to ask questions more in depth, like how exactly do humans regulate line one? Meaning, like, if we look into these restriction factors, what parts of them interact with line one? What is important about these restriction factors? Do they have something in common? Are they part of our immune system or not? And then we can also use experimental evolution to try to tackle some of these questions. And this is one of the things that I really like about this lab, is that we can ask the question, what if we change that interaction? What if we change that the molecular details of that to resemble what happens a million years ago? And we can also try to like change that interaction to see what's going to happen in the future. Are we, as a species, as a human species, going to be able to maintain control of this pathogen, or at some point this pathogen that it's hanging around in our genomes is going to be able to evade and like go loose? So we use in our lab these mainly these four fields, in these four methodologies to target to address these questions. And I want to talk to you now about an experiment that I did, trying to understand more about how line one control happens in our cells. So I took 4 different line ones that I named A, B, C, and D. And these line ones are present in our genomes. And I put them in cell line 1 and cell line 2. I did that with line 1A. I put that line 1A in cell line 1 and cell line 2, and then I measured the jumping activity of that line 1. And then I did the same for B. I put line 1B in cell line 1 and cell line 2 and measure the activity. And the same for C and the same for D. And what I found was that there were 2 line ones, A and C, that were pretty actively jumping. They're considered to be active in cell line 1 and in cell line 2. But we found a huge difference in how line 1B was behaving. In cell line 1, line 1B, it's dead, or it has almost 0 activity. While in cell line 2, that line 1B, it's pretty active. What that suggests is that there's a context. There's something happening in cell line 2 that is not present in cell line 1. And therefore, line 1B can evade or can be active in cell line 2. This is something that we have learned about these line ones, and we're looking forward to expand this into other cell lines, other species, and trying to understand more about what makes line 1 capable of evade host restriction, and how does that relate? Why is that relevant and important to understand line 1 evolution and in the human genome? So there are other really cool things that transposons do. One of them is that there are other transposons that are not line 1 in our genome, and they use line 1 machinery to be able to access your genes. Like these other transposable elements, because they're not autonomous, they have to hitchhike line 1 machinery, which is pretty interesting phenomena. The other thing about transposons that is really cool too is that coevolution does not necessarily mean conflict. There are parts of our genomes that are proteins that we have domesticated, that we have adapted to work in our favor. there's one example of a DNA transposon derived gene that it's really essential for our immune system, and that gene is called RAG, so that we can use we can use transposable elements as prime material to create new genes over big evolutionary times, of course. And the other cool thing that I think is super amazing is that transposon activity has been shown to be key for neural development and like oocyte formations. And these are processes that happen during like embryonic development, but you would think that like the narrative that I just gave you is like, well, these transposable elements are bad because they can generate disease. But they have also shown that if you have activity of line one, if you have activity of other transposable elements in the brain, they're really important for making for cell differentiation, like for stem cells differentiation into like neurons and other types of cells. And also that happens in these cells, sperms are eggs too. So that's pretty cool. Okay, so I'm gonna have a summary of what I just told you. We talk about how half of our genomes and like more it's made of transposable elements, that line one retrotransposons are the only measurably active and the most successful family of Ts in humans, and therefore it's really relevant to understand how they evolve and how they behave in our cells, and how in our lab we try to understand the evolutionary of line one and human, because it's essential for us to determine how they drive disease and how in the future they can how they drive the disease, and how we treat those diseases, and how we as humans are going to be able to control them or not. So I think that was most of it. I want to say gracias to Janine and everybody here, and I want to thank my program, the MCB program, and the McLaughlin Lab for supporting me, and the CMB grant. Feel free to add me on Twitter, and now I will respond to questions if you have some. Please do.
All right, thank you so much. And that was there's a lot of interesting things put into that presentation, and I'd like to invite participants to go ahead and add some questions into the chat, and we'll go from there. So the first one is, does L1 have a target sequence for its insertion site?
Yes, it's, give me a second, let me go to the start. I don't know how to, okay. Yes, so, yeah, they do. It's a lot of T's and A's. I don't know the sequence itself in my mind, but I know it's not. It's pretty generical, but we know that they are enriched in places that they have a lot of T's and A's.
How does cutting out DNA work? What's left behind, and how does it choose where it goes? Which I think is a little bit related to the question before, but can you talk about how the cutting out works? What's left behind?
So this differs between transposons. So if we talk about DNA transposons, so they have at the end of these sequences, they have some repeats, and because the DNA in our cells, it's super folded. These repeats sometimes are able to recombine, and there are proteins that help that facilitate that process of recombine and like make a loop and excise out that part of the DNA, leaving behind one of the repeats that were in the site. So that's what you see when, or that's this block of here, this black blocks that you see when DNAs are cut and pasted. In case of the retrotransposons, there's also this sequence that you can find in the insertion sites, but because these are not taken out, like there's no empty space that you can find. Sometimes when retrotransposons are inserted into a new place, that process has really low efficiency. So the insertion can be a truncation, can be half or part of the transposon itself, and that in some ways that it's represented here as blue, like mutations or like truncations, and that retrotransposon can't jump again because of that.
So just to be clear too, because this is not something I was clear on when I heard this the first time. So line 1 is the retrotransposon.
Yes.
Yeah. So it's just to it's in that class of transposons. I was really interested in how we regulate line 1. Like, what do we do? What kind of things does our genome do to regulate line 1?
Yeah, so there are proteins that for restriction factors, and they do a lot. They can literally target any point of this cycle. For example, they can, in order for line 1 to be expressed, you need you need a machinery that initiates this reading. And if you have a factor that it's close to line 1, the sequence in the genome, then you can prevent other machineries to jump in and try to read. There are other ways to do that, like attaching to line 1 mRNA here. If you attach to line 1 mRNA and stop translation to proteins from going, then you're not going to have line 1 reinsertion. So there are many factors that have been associated with line 1, with researching line 1. Cool thing about our research is that even if in literature you can find all these factors, there's only a few examples of restriction factors that have been evolving to satisfy the hypothesis that I showed to you. That we know that line 1, we have a lot of line 1s in our genome. We have like 500,000 copies of these parasites.
That was another question I had, was like, how many? Yeah, 500,000 copies. It's amazing.
Yes. And some of those are truncations, like not all of them are able to jump. Yeah. But that is pretty much like, that's in terms of percent, that is like 17%. So what was I gonna say? Yeah, so what we're looking for is what is driving this evolution? What is driving line 1 to change? Yeah. And it's really hard to find proteins in the host that are also showing signatures of being evolving or co-evolving in this relationship, because these other restriction factors, these defense mechanisms that we have in our host, they don't only sometimes like target line 1, they can also target other viruses, other parasites, other transposable elements. So it's harder to see evidence of evolutions against one transposable element.
Yeah. And there are some, like you said, there's some benefits too to having, right, the system. So,
yeah. Well, it depends on the cell.
yeah.
So in neurons, this is I didn't talk about this, but line one has a promoter, and this genes that let me see. Okay, promoter sequence is this place where the machinery for reading lands and then allows for reading the DNA. So if line one jumps into a place where there was no promoter, then it will activate reading of genes that are nearby, and therefore start producing proteins that in that cell were not produced before. So in that way, you can think how cells or like stem cells can differentiate into like brain cells or heart cells or this or that based on the proteins that are expressed um, because the uh, because when line one jumps or like transposable element jumps, it changed the genetic program. It changed how things are expressed or how things are read.
That's really interesting. We have a couple more questions in the chat. The first one is L1 autonomous in other model organisms, and what other organisms are being studied to understand the activity?
Yes. So L1 as a yeah, as a transposable element is autonomous in other organisms as well, but those other organisms have other TEs that can be active and autonomous too. Not autonomous though. So you can pretty much see line one research from yeast to humans, to like most eukaryotic cells have a version of line one. So it's yeah, in line one as a long interspersed element one, and has evolved more really specifically in mammals too.
And so the second part, are other organisms, actively being studied to understand line one activity? Are there like specific model organisms for studying this activity?
Yeah. So fish, a lot of reptiles. There are a lot of studies of line one and other repetitive elements in this model, and it's really great that that they're there, because then we can compare the patterns of evolution. For example, in fish, there are multiple families of line one coexisting. Like imagine that there's like line one. Well, yeah, there are multiple families of line one coexisting. But in humans, there's this pattern that a line one becomes active and create all this family, and one of those line ones becomes active and create another huge family. Like there's only one family that's always active or that's always like present, and that's more evidence that there's something selecting these line ones in humans that is not present in fish, for example, where multiple families of line ones are coexisting, and there's not that pattern of only one succeeding the previous family.
Yeah, it's really, I think, the picture often that you see presented in textbooks or schools is the genome being really fixed, and just to understand how dynamic it is really interesting. So another question, is anything known about the differences in these restriction factors between the cell lines that you were studying? So when you're seeing the difference in cell lines, the questions about this. Yeah.
So in this particular case, I didn't brought all the details because I think I mentioned a lot of details before in the presentation and didn't want to overwhelm. But in this case, cell line 1 is a human cell line and cell line 2 is a cat cell line. So cats have definitely different restriction factors than humans. So we think that the difference that we can see in line 1B versus other line 1s that are present in this data set, this graphic, is that there's a restriction factor in line 1 that is not allowing that line 1 to jump. But in cats, it's not present and therefore is active. That's our hypothesis.
Mm-hmm. Great.
Can I ask a quick question about the cats?
Yeah.
So is it not active in cats because it's not historically there, if that makes sense, like in cat genomes?
Yes. So we don't know which restriction factor it's acting or is it influencing the jumping activity itself. But like this suggests that it is a restriction factor, that is a cellular difference. But I have no information about what specifically is targeting it. However, we know that is really specific to line 1B because when we do phylogenetics and we look at the relationships of A, B, C, and D, they're closely related. They have a few like dozens differences in the sequence. So it has to be something really specific.
Yeah, that was kind of related to the next question. How different is A, B, C, and D? Did you expect none of them to be restricted in cats?
So there were two hypotheses about what will happen in cats. First, like I was like, well, I wanted to test the activity of these line 1s in humans, and I want to try to find out how cellular differences are relevant. But because these line 1s are human line 1s, the possibility that everything was active or nothing was active was there. Like these line 1s could need other factors that are specific to humans in order to jump. And that was a risk we took. But then when we look at the data, we see that, for example, D is not completely active in cats. It's not super active in cats, meaning that there's still differences, and they can still like this model does give us some proof of concept experiment where we can see the line 1B is targeted.
One of the things that we had talked about was that a lot of science teachers who do biotechnology might do like an ALU type of activity to look at ALU repeats in some human samples, perhaps student samples either. And so can you talk a little bit about the relationship between something like line 1 and the ALUs?
Yes. So the cool thing about other transposable elements is that they are significantly shorter. So sines, ALUs, and other transposable elements that are mobilized through line 1, they have like 300, 200 base pairs. Line 1 has 6,000, and this is why working with line 1 in the lab or like you doing molecular biology with it, it's so complicated. But yeah, doing ALU and sines is really cool and easy way to like approach transposable elements from the teaching point of view, because you can do really easy experiments, in a lab-based class.
So the ALUs are using the machinery from the line to move.
Yes.
And do all non-autonomous ones use line machinery, or are there some that use other kinds of machinery?
Yeah, most of them. I don't want to say all. I think it's all, but most of them.
Yeah.
Yes. This is another really interesting thing that we are exploring in our lab is that the evolution pattern that we see in line one could be driven by the host, but it also could be driven by the competition with other transposable elements for the machinery. So if you are transposable elements that if you're young and you have an older brother or your sister and you want to go out, you're gonna try to catch a ride, right? So these other transposable elements that are shorter and that don't have the machinery to jump, they might competing and creating um, pressure for these line one to be further specialized for themselves.
We have time for a couple more questions. Invite students or any other teachers if you have questions. You can unmute yourself if that's easier than typing in the chat, or you can type in the chat.
I can, sorry.
And I have a couple more too. You mentioned L1 driven diseases. Like what kind of diseases are driven by L1?
I can mention from the top of my mind that there some genetic syndromes.
But they're pretty rare.
In a lot of cancers.
Oh, and a lot of cancers too.
Yes. So mostly because of reprogramming. So if line one lands in a place where there's an oncogene that has been silent, an oncogene being a gene that promotes cancer, and then it changes from silent to being readable and active, then it will drive the disease. Or yeah, most of the examples that I've seen in literature are related to cancer and other genetic syndromes. Yeah.
Oh, is there homology to any viruses?
Yes. So this is really interesting because viruses, they can exit the cell. So most of the time, well, most viruses, but most of the time viruses can spread horizontally. They can spread to other members of the population. In this case, this is a virus-like element, but it only spreads from mother to the offspring. So these transposable elements are inherited vertically, are transmitted vertically, and that changes a lot the way we study how they evolve. They're not as in some ways they're not as urgent as a virus, as a pandemic, for example. Yeah. But they're still really important in if you have a line one driven insertion, line one disease, line one that is drive disease, and that is inherited in our population for a long time, then you have a problem there.
Okay, last question here. I think you said there were 12 differences between some of the L1s you used. In general, how divergent are L1s?
Well, not 12, they're like dozens, meaning like, so, okay, so if I show you this, there more than 20 between all of them. Like the answer is different if you compare A to B or A to C or A to D, but they're really similar considering that they're 6,000 bases long, and with only like dozens of mutations. And the other part of the question was
Oh,
how,
so
okay,
in
yeah,
general, like how in
in
general, if you
general,
look across all the line ones.
yeah, you, there's so many
Yes.
polymorphisms, so many mutations that you can have.
Yeah.
Um, the only things that you need that are essential are those two genes and having that promoter region. Like in some ways, those two genes can change, um, and that's what we're trying to figure out. Like what changes are related to more activity or to less activity. Um, it's still really young question in the field.
Okay, thank you very much. Can we give Ricky some applause and thank yous? You can unmute yourself if you like.
Thank you.
And I'm going to switch my
Thank you
screen
for
back
being
on
here.
just for a second and can wrap this up. So thank you so much. That was fascinating to learn about line one. Next week, we're doing something a little bit different. And so let me get that sharing. We're going to hear the journeys of a few people in science. And so these are the people who will be sharing some of their journeys. They have different kind of positions. A few of them are graduate students. There's a lab tech. There's someone who has finished his PhD and is working in a lab. And so there are bios and the opportunity to submit questions in advance. And those are linked in the chat, but they're also on our webpage where all the events are listed. And you can also get the recordings there. So the bios and opportunities for submitting questions are on, they live on this page. I'll remind you also that July 7th, we have no Hutch at Home, but we are now booking through August. So let's make sure to keep spreading the word, and I'm really excited to see all of you hopefully at the panel next week. And now I will open it up if there's any more technical questions for Ricky. He's agreed to stay on for a few more minutes. And if there's something you want to dive in a little bit deeper, we can do that now. But I thank all of you for coming today. And thanks again, Ricky. It was an amazing presentation and beautiful slides. The feedback form is here too. I've also pasted that into the chat, and the URL is there if that's easier for you than taking a snap of the QR code. But yeah, so thanks everybody. And any additional questions for Ricky at this point? You can feel free to unmute yourself. You good? I think you have answered the questions. I could keep going, but I'm not going to keep you here. Thank you though for, it's really fascinating. I mean, I do think the value for students a lot is thinking about how dynamic the genome is. Really, it's just amazing, you know. And that's not something we really cover very much usually. So just how much it's changeable.
I really enjoyed the opportunity to talk. Thank you.
Thank you, Ricky.
Thank you, Ricky.
Ricky.